So what you're looking at behind me are bacteria swimming around inside the guts of a live zebrafish. This movie my students and I in my lab took a few months ago, and it raises a lot of interesting questions. So first of all, you might be wondering, is this going on in my own intestines? And the answer, frighteningly enough, is yes. You also might be wondering, why would anybody want to look at bacteria inside fish guts? And that's a really important question. I'm going to come back to that in a little bit. But finally, you might also be wondering, why is a physicist looking at bacteria in fish guts? I mean, aren't you people supposed to be looking at lasers and magnets and particle accelerators and stuff like that? What's going on with the fish guts? So it's that question that I especially want to talk about in the next few minutes, because it's one that I think really illustrates what's particularly unusual and exciting about 21st century science. So we think about particular fields of science like chemistry and biology and physics and geography and things like that. And we also partition our teaching of science into these compartments. And that's not a bad thing. The universe is a really complicated place, and if we didn't impose some structure on it, we'd be hopelessly lost. But it's important to keep in mind that these compartments, these fields, are artificial. Nature doesn't care what's chemistry and what's physics. These distinctions are ones that we create ourselves. And it's also important to keep in mind when we create these distinctions that very interesting things might be going on at the boundaries or the intersections of these particular fields. And the intersection that I've gotten really, really fascinated by is the intersection of physics and biology. And what I've been wondering for quite a while now is, are there interesting things about life or about nature in general that we learn from studying this intersection of biology and physics that we wouldn't get just from studying physics or biology alone? So it turns out, not surprisingly, since I'm standing here, the answer to that question is yes, there are lots of interesting things there. And moreover, this biophysical perspective tells us a lot about how life works, both at macroscopic scales, the scales of humans and animals and trees and things like that, and also microscopic scales. Now, the fact that physics influences how life works at macroscopic scales is fairly obvious. I mean, nobody would be very surprised if I told them that, like, the flying of birds involves principles of aerodynamics. But it turns out to be just as important and just as universal that physics influences how life works at microscopic scales also. In fact, the more you look into this, the more evident it becomes that essentially all biological phenomena are in some way inherently physical. So for example, one thing that's going on inside you right now are that some of your cells are moving around. You have, for example, immune cells that are extending little finger-like projections and pulling themselves, crawling through different tissues as they patrol your body. You have proteins. So a protein is actually a long chain-like molecule. You have proteins that bend and fold themselves into particular shapes, and those shapes help them do particular functions. This example I'm showing up here is actually a protein called cholera toxin, which not surprisingly is involved in cholera, that folds itself into a pore that ends up allowing things to leak out of your cell membranes, which is not a good thing. So you have DNA. Inside every single one of your cells, you have a nucleus. That nucleus is about 1/millionth of a meter wide. And inside every one of those cell nuclei, you have a meter-long strand of DNA. Okay? So that DNA has to be bent and packaged into that very small space. And it turns out that the mechanics of that, how the DNA ends up getting wound around little protein spools, is not only really fascinating, but also influences how and even whether particular genes get turned on. So for all of these sorts of things, one can ask, how do physical things like energies and forces manifest themselves? How do the material properties of these biological materials influence how they work? So in my research lab, my students and I are particularly concerned with one class of biological materials called cell membranes. So as you probably know, at the boundary of all of your cells, and it turns out at the boundary of all the organelles within your cells, you have membranes. So these membranes are biologically very important. They're in fact more than just boundaries. A lot happens there. Actually over 1/3 of the human genome encodes proteins that are located at membranes. And something like 70% of pharmaceutical drug targets target activities that go on at membranes. So membranes are biologically very important, but they're also physically really fascinating materials. They're essentially two-dimensional. They're just a couple of nanometers thick. That's a few billionths of a meter. They're fluid, meaning that the molecules in the membrane, including these proteins that I illustrated here, are free to move around in this two-dimensional liquid world and interact with one another and build structures and perform chemical functions. And they're also flexible. The whole membrane can bend and curve in 3 dimensions as cells go about the tasks they want to perform. So all of these physical properties, it turns out, are made possible by an underlying architecture that all membranes, whether in you or animals or even plants, share, that of a so-called lipid bilayer, where there are these little molecules called lipids that I'll say more about in just a moment, colored in blue here, that together make up the basic architectural element of all membranes. So these lipids, they're important, they end up forming membranes. And so you might wonder, well, we hear a lot about genes these days. What genes direct the assembly of lipids into membranes? So what genes tell lipids to assemble into membranes? And the answer to that question is none. It turns out these lipid molecules are just themselves physically very interesting. Each lipid molecule has what's called a head here that likes water. And each lipid molecule has these greasy, fatty tails that don't like water. Very much like oil and water will separate, these greasy tails will demix from water. So if you just take a bunch of lipid molecules and just toss them into a beaker of water, they'll spontaneously assemble themselves into this 2-molecule thick membrane just to shield these these water disliking tails from water. I'm trying to avoid jargon as much as I can. So. I might slip once or twice. So lipids will just spontaneously assemble into membranes to shield these greasy tails from water. So there isn't any gene that has to encode this. Your body just creates the lipid molecules and they assemble themselves into membranes just from these physical interactions. Now that's kind of cute for membranes, but it's also a very deep kind of lesson, because there's a fundamentally interesting fact that we've learned about genes in the last 20 years or so. So if you look at yourself, you're a complicated thing. You have hundreds of trillions of cells. Each of those is doing countless tasks, countless chemical reactions, countless modes of growth. You're forming patterns, you're building structures, you're doing all of this stuff, and you're doing it all with just about 25,000 genes. That's the content of the human genome, 25,000 instructions that your DNA is encoding. So how do you go from 25,000 genes to all this complexity that you see in yourself? Now, that general question is a really, really deep one that drives a lot of inquiry these days. And very roughly speaking, there's 2 parts to the answer to that we're figuring out. One that I actually won't really talk about is that genes interact in networks, and combinations of genes can do things that individual genes cannot, and through combinations you can get a lot of interesting phenomena to occur. But the other thing is that physical processes play a really large role in biology. And just like the lipids I showed you assemble themselves into membranes due to physical interactions, it turns out a lot of things that go on in organisms involve self-assembly driven by physical interactions. So to understand life, we need to understand biophysics. So to give you a little bit more concrete an example of what the sort of things we do in my lab are, I thought I'd tell you a little bit about an experiment that we've been doing recently. So this again has to do with membranes, and one of the things that membranes are involved with is trafficking stuff from one place to another in your cells, or from the inside of your cells to the outside. So things like neurotransmitters being released at your neurons, or proteins being created at one part of your cell and moved to another part, are often transported by encapsulating them in little membrane-bound capsules and ferrying those capsules to their destination. So it turns out that there are proteins that grab onto membranes, bend and curve them into these little capsules, pinch them off, and then ferry them to wherever they need to go. So we know a lot. So these trafficking processes are really important to cell function, and also problems with trafficking play a large role in a lot of diseases. So because of that, a lot of people study trafficking. We know a lot about what proteins are involved in these processes. We know their names, their structures, things like that. But we don't have a great sense of what they actually do, what they mechanically do. So to get at this, we decided to do some experiments asking whether the proteins that are involved in these sorts of trafficking things, the making of these capsules, can actually alter the mechanical properties of a membrane. And specifically, we wanted to know, well, for various reasons, we hypothesized that maybe they might alter the stiffness of this 2-dimensional membrane sheet. So conceptually, a really easy way to study that would be to take a membrane, grab it, pull it, and see how it responds to tell how stiff it is. Now, it turns out we can actually do that. There is a tool that was developed in the past couple of decades called an optical trap that allows you to take laser light and use focused laser light to apply forces at microscopic scales. So we can take membranes, attach things like little beads. This bead here is about a few millionths of a meter in size. We can attach little beads to the membrane, use our optical trap, that's the thing in red there, to grab onto the bead, pull on the membrane, thereby creating these little tethers of membrane. And then, as you can see from the movie here, we can let go and watch the membrane pull the bead back. And from that, watching how the membrane pulls the bead back, we can tell how stiff the membrane is. Basically, the stiffer the membrane is, the bigger that force is going to be, as you might guess. So doing this, we can measure the rigidity, the stiffness of the membrane, and we can add in proteins that are involved in these trafficking processes. And we picked a particular one that ends up being important in trafficking stuff from the endoplasmic reticulum. That's, I guess, jargon word number 2. It's one of the organelles in your cells. And we can measure the rigidity of the membrane as a function of how much of this trafficking protein there is. And what we discovered is that the more of this protein you add in, the softer the membrane becomes. It becomes less and less rigid. And it's not even a subtle effect. If we add enough of this protein, the membrane rigidity can go pretty much to 0. It just becomes extremely easy to bend and deform. And this was really exciting for us because it turned out it was the first time that anybody had shown that any trafficking protein can lower the rigidity of a membrane, which we were excited about. So what we hope is that this brings us closer and closer towards actual predictive mechanistic models of how processes like trafficking work, and then, of course, how they can go wrong. The other thing, though, that we're kind of excited about is we're trying to use these proteins and membranes to artificially create little capsules and little membrane-encapsulated packets, basically mimicking biology for artificial material-related ends. So biophysics gives us a really broad perspective on how to think about life. And most of what we do has involved, in my lab, has involved things like these membranes. But recently, in the last couple of years, we've also branched out to another realm that we think is very fruitful for biophysical perspectives. And that is, how do collections of cells organize themselves into structures? And is there, can we tell something about the mechanisms behind that? And as an example, these are my 2 kids. They managed to assemble themselves into this, these wonderful arrangements of trillions and trillions of cells, starting out just from 1 cell each. And how does this structure and organization happen? So there are a lot of different tissues and organs that one could think about and try to investigate. We got especially excited by something that you wouldn't normally think about as an organ, and that is the collection of microbes that exist in your body, especially in your gut. So it turns out that you are actually mostly microbe. You have about 10 times as many bacterial cells as you do human cells. Thankfully, they're smaller than the human cells, so you win by volume. But if it were put up to a vote, if you voted, you would lose. Okay. So most of these bacterial cells actually reside in your gut. And not only do you have lots of cells, you actually have lots of different species of bacteria. And it's been increasingly realized in the past few years that these microbes play enormously important roles in a lot of your physiological processes. Things like immune response, digestion, even how cells differentiate into different types of cells. Also, it's been realized that differences in microbial composition are very strongly correlated with a lot of really interesting diseases, and very interesting and very important diseases. Things like diabetes, inflammatory bowel disease, even obesity are strongly correlated with microbial composition. So it turns out that we understand very little about how these microbial communities assemble, especially at sort of the very early stages. Like you in the womb actually didn't have gut microbes. It was as you were being born and then after you were born that these communities started to develop. And we don't understand this process well at all. And there are things that are very hard to study in humans. You can't just you don't have a transparent belly that we can look into and see your gut microbes. And it's, I think, a very good thing that you don't. So how can we start to study this? Well, we actually make use of a model organism called the zebrafish, which was actually really developed and popularized as a model organism here. They're beautiful little fish, and one thing that's very nice about them is that at early stages of development, they're transparent, so we can look inside them. And so we build actually new types of microscopes that allow us to do 3-dimensional imaging of developing zebrafish and see what's going on there. So these are 2 of my graduate students. The one in front here, Mike Taormina, is actually a native Oregonian who grew up in far eastern Oregon. He's a graduate student here now, and he was actually also an undergraduate here in the Honors College. So we built this microscope to look at these bacteria inside zebrafish. We also make use of the fact that we have wonderful colleagues in biology who could do things like engineer bacteria with fluorescent probes in them so we can see them glowing and visualize them and start to learn things about them. So doing that, we can do things like watch these bacteria swimming around and get some idea of how motile they are and how they can explore different parts of the gut. We can also do things like take these three-dimensional movies for many, many hours and watch what initially starts out as a very small population of bacteria, just a handful, you can count them on one or two hands, watch how that divides and expands into populations of tens of thousands. And we can also see, do they form particular colonies? Do they form particular colony sizes? How do they interact with other bacteria and create sort of a functioning ecosystem in this animal? So doing this, there's a lot to explore, and we're really interested in studying what are the shapes and sizes and structures of these microbial communities that exist in zebrafish, but also exist in very similar ways in humans and actually all other animals. And again, what we want to do is create predictive models of how these communities assemble, and then how they respond to perturbations like antibiotic treatments and other sorts of things that affect the microbial communities. So I do all this with the students, both undergraduates and graduate students who work in my lab. About half of these are undergraduates, half of them are graduate students. And one thing that's especially nice, one thing I'm especially happy about with my group is that these students are in physics, chemistry, and biology. So they bring a very wide range of perspectives to the lab that leads to a lot of really stimulating discussion, and it's just a lot of fun. So hopefully I've convinced you that understanding physical interactions and mechanics and assembly gives us a really deep perspective on understanding life. And also just that there's a lot of really interesting things to see at this intersection of physics and biology. So, thank you.